All right. Okay, guys. So tomorrow in lecture we will go over the syllabus. We'll kind of do an introduction into the. Okay. We'll do an introduction of kind of what it is, stalker feedlot management, some things that need to be considered around budgeting. So I would say overall this class is kind of a healthy mixture between nutrition, econ, and animal health. Hal, would you add anything to that? It covers it pretty well. Okay. So today we're going to do a feeds and feeding review. Okay, so this should be a review. Some of this information may be new, okay, or some of this information may be in a much greater depth than what you guys did in feeds and feeding. Okay, but it's kind of a baseline knowledge of nutrition that you're going to need to do well in these subsequent lectures in the class. Okay. All right, first, scan this QR code. You can use your phone. It's going to take you to a Google form. Just want to gauge kind of everyone's baseline knowledge of some concepts around nutrition. We're missing 4.
So in terms of group,
2 more.
at least according to campus.
Okay, we've got 3 responses so far. 4. 5. All right, I'll start wrapping it up. Anybody still working on it? All done? Okay, I'll take a look at how everybody did. It's not for a grade or anything, but all right. So I'll pretty well start all my lectures, lab lectures, whatever it is, with some learning objectives. So in terms of reviewing for exams, they're probably a good place to start. Make sure that you know these learning objectives that are listed at the beginning of each slide deck. Okay. So for today, I want you to understand, the differences in nutrient characteristics. And this goes throughout 95% of the class. I don't want you to memorize equations, but I want you to know how to employ them. Okay, most equations I will give to you as like a cheat sheet for exams or some of the exams, the math portions that we'll have undergrads do in the lab. So we'll do that before our exam reviews. Okay, so I'll give you a printout document with different equations on them that you can use in the test, but it's not going to help you if you don't know how to employ those equations. Okay, but in the real world, you're not going to have to memorize equations, but you will have to know where to find information and how to use them. Does that make sense? Okay. Today, I want you to know what the different fiber fractions make up NDF and ADF. Okay. So I actually taught honors section of 318 last semester, so I know that you guys learned this in feeds and feeding. Okay. I want you to know how the NE system works. We're going to go a little bit more in depth on this today than what you would have covered in feeds and feeding. Okay. And then, for example, if we give you a certain level of dry matter intake and the energy content of that diet in terms of NEM and NEG, okay, I want you to be able to calculate a projected average daily gain from those numbers. Okay. Conversely, if we give you uh, so much megacals of or so much kilograms of dry matter intake. Did I say that? Mm-hmm. Okay, then if I give you a average daily gain that animal has had, I want you to be able to calculate how much dry matter intake that animal must have consumed to meet those net energy for maintenance and gain requirements. Make sense? So we'll go over that today. Finally, we're going to touch on crude protein. We will go into more depth than what you would have gone into in feeds and feeding on this, but I want you to understand what the fates of crude protein is for ruminant animals. Okay, RDP, RUP, microbial crude protein is MCP, TDN, allowable MCP synthesis. Okay, I don't think you would have gone over that in feeds. Maybe I'm wrong. And then ultimately metabolizable protein. Questions so far? Okay. All right, proximate analysis. This is something you guys went over in feeds and feeding. Okay. These are abbreviations I'll use in this lecture. Okay, so dry matter is a percent of as fed. Okay. Organic matter is a percent of dry matter. So it's 100 minus ash content. Ash content represents minerals. Okay. Neutral detergent fiber, NDF, ADF, acid detergent fiber. We'll talk about lignin some, but we can measure that using acid detergent lignin. Okay. Ether extract is our estimate of fat. Crude protein. Okay. Need to know how we calculate crude protein. So what do we actually measure when we're estimating a diet's crude protein? Who remembers? Anyone remember? Nitrogen. And then how do we go from nitrogen to crude protein? What's that? No. nitrogen times something. 6.25. Why 6.25? Okay, so 6.25 because 1 divided by 0.16 is 6.25. We assume that all amino acids are 16% nitrogen. Okay, so that's something you would have learned in feeds and feeding. When did you guys have feeds and feeding last? Been a year? Okay. All right. So let's remember some of these things. Okay, so neutral detergent, insoluble crude protein. So that'll be the nitrogen content of the NDF residue. Okay. Acid detergent, insoluble crude protein would be the nitrogen content of the acid detergent fiber residue. Okay. TDN is total digestible nutrients. So I think one of the questions in that quiz was, what is an estimate of energy? Okay, you guys put TDN, some of you put net energy for maintenance, some of net energy for gain, maybe, hopefully. Okay. Cool. Questions? All right. Okay, so dry matter, organic matter, and ash. All right, so we can weigh the sample as is. We can dry it in an oven somewhere between 50 to 100 degrees Celsius for 48 to 72 hours. Really, we should take it out periodically and stop drying it whenever the weight doesn't change. So when that sample doesn't lose any more additional water, okay? So sample that's left divided by the sample that we put into the oven times 100 gives us dry matter percent, okay? We can take that sample, put it into an ashing oven. So we'll ash that sample 500 to 600 degrees Celsius for 2 to 6 hours. All the organic matter, so non-minerals, will combust. What you're left with is ash. Okay, so that's an estimate of mineral content. Okay, so 100% minus ash tells us organic matter. Okay. Okay. Took this picture right out of the beef NASM. You guys would have seen that book in Feeds and Feeding. Okay. So, losing my mouse. Okay. So here we have cell contents that are non-fibrous. Okay. So they're not going to be contained in the NDF fraction. Okay. Then we have parts of the cell that are contained in the cell wall. So those would be fiber carbohydrates. Okay. All fiber carbohydrates, hemicellulose, cellulose, and lignin are contained in NDF. Only cellulose and lignin are contained in ADF. You guys remember that? Okay. Hopefully we get to talking more together and this is more interactive as we get more comfortable with each other and the semester goes on. So how do we measure NDF? No, you guys did this in like your first feeds and feeding lab, right? We can take these filter bags, this F57 bag, load a half a gram of sample into it. Okay, we know how much sample we put into that bag, seal it, put it in this fiber analyzer machine, boil it in either neutral detergent solution or acid detergent solution. Okay, if we're doing NDF or ADF, whatever gets washed out is non-fibrous. Whatever remains is fiber. So we're trying to measure the residue. Did you guys find those slides? I'm seeing people actually taking notes on paper. I put the slides in Canvas. You guys have those? If you'd rather write it on paper, that's fine. But if you want the slides to take notes on, they are on Canvas. Good. Okay. All right. So NDF, hemicellulose, cellulose, lignin, ADF, just cellulose and lignin. So we take that sample, put it in a bag, boil it in NDS, neutral detergent solution, or ADS, acid detergent solution, and whatever is remaining is either NDF or ADF. Okay. So we express that residue as a percent of the total sample, and that gives us percent NDF, percent ADF. Okay. After we do the acid detergent solution, we can incubate those samples in 72% sulfuric acid at ambient temperatures. We typically will do that in, this is called a daisy. After 3 hours, we remove those samples, dry them, weigh the residue, and whatever's left is lignin. So acid detergent lignin, express that as a percent of the total sample. That gives you lignin as a percent of dry matter. Make sense? So would a higher quality feed have more or less lignin? Less. Would a higher quality feed have more or less NDF? Less, right? Okay. So typically our finishing diets will be somewhere around 20% neutral detergent fiber. Grower diet could be somewhere in 30s. Pastures somewhere, depending on the how high of quality it is, somewhere 40 plus. I think Kyle's study was 60-ish. Okay. All right, fat content. So there is a bag, filter bag technique for ether extract measuring. Okay, but really we take that sample, we boil it in ether. That ether solubilizes the fat and makes it evaporate. Okay, so what is left after boiling it in ether is fat-free. Okay, so we figure out how much of that sample was left or lost out of the bag after boiling it in ether, divide it by total amount of sample that was incubated, times 100, gives us ether extract content. Okay, so this is an XT15 fat analyzer. These are those XT4 bags, same company as these F57 bags. Very similar concept on how to measure it, except in this case, we're not interested in what's left in the bag. We're interested in what was lost. Make sense? Okay. Protein. Okay. So as we said, when we're talking about crude protein, we are really talking about nitrogen. Okay. We assume, and this is probably more true for ruminants, that that nitrogen can be utilized by the animal to create amino acids, really not utilized by that animal, utilized by the microbes to create amino acids that that animal can use for metabolism. Okay, so we can measure crude protein as nitrogen content times 6.25. Again, 6.25 is because we assume that all amino acids are 16% crude protein. Okay, so 1 divided by 0.16 is 6.25. There's really two ways that we can measure nitrogen. One is through the Kate-Hall method. Okay, so we digest the sample in acid. That digestion converts all that nitrogen into ammonia, and then we measure ammonia concentration. Okay. The other way, and probably what's most commonly done today, is the Dumas combustion method. So we use these machines. This is a Vario Max carbon nitrogen analyzer. We just bought one that we have in my lab that's a LECO. They do the same thing. You put the sample in, it combusts that sample. All that sample is converted to gas, and it goes through different tubes that convert all that nitrogen gas, so into nitrous oxide. Then it goes to an analyzer to measure nitrous oxide content, and it does that to calculate nitrogen. Does that make sense? So if we take the residue after the neutral detergent solution soak or the acid detergent solution soak, and then combust those or analyze those for nitrogen, we get neutral detergent insoluble crude protein and then acid detergent insoluble crude protein. Make sense? So if we have more nitrogen that is found in the fiber fractions, would we expect that nitrogen to be more or less digestible. You have more that's in the fiber fractions. Do we expect more of that to be available to the animal or less? Less. Okay. So I know this was covered in feeds. They have adjusted crude protein. If X% of your crude protein, I don't remember the exact number that Dr. Parsons would say, but if X% of your crude protein is found in acid detergent, as acid detergent insoluble crude protein, you need to make an adjustment to your crude protein content because less of that total crude protein will be available for the animal. So if you guys remember from your NEARS report, you have total crude protein, and then there was an ACP, so adjusted crude protein. Okay. Energy. I know for a fact this is reviewed for you guys. Okay. But I'm very fond of questions that talk about the energy flow diagram. So don't be surprised if you see this later. Okay. So we take that sample and we can do what to it to measure gross energy? How do we measure gross energy in a feed sample? We call it heat of combustion. So we can put it through a bomb calorimeter. You guys remember that from feeds? Okay. Put it in a bomb calorimeter. Most of those bomb calorimeters are called adiabatic. Okay. What that really means is the combustion chamber is housed and it's surrounded by water. You blow up that sample that produces heat, and then all it's really measuring is the temperature change of that water surrounding that combustion vessel. You relate it to a known calibration curve, and you can measure kcals per kilogram of feed. Okay? So we feed that to an animal, and that animal is only going to digest a fraction of that. So some of that feed is going to be excreted in feces. Some of that feed energy will be lost as feces, so fecal energy. So gross energy minus fecal energy is digestible energy. Okay. Digestible energy and TDN should be highly related, right? 4.401 times DE should be TDN or something like that. We've got it in some slides down here. Okay. So of that DE, of that energy that was digested, some will be lost as methane energy, gaseous energy losses. So would we expect that gaseous energy loss would be higher or lower in a stalker animal versus a finisher animal? Which system will produce more methane?
Stalker.
Stalker. Okay. Higher forage diets will produce more methane, will have more energy loss as methane than lower forage diets. Okay. So we say that gross energy or methane energy loss as a percent of gross energy is somewhere in the range of 2 to 12%. So it'll be on the other end, the 12% in on high forage diets. It'll be at the lower end on low forage diets. Okay. Typically, we use 3% of gross energy intake is lost as methane energy in a feedlot. Okay. All right. So some of that digestible energy will not actually be metabolized in the animal, right? So it's going to be excreted as urine energy. Most of that urine energy is nitrogen excretion. So conversion of gross energy or it uses some energy to convert excess nitrogen into urea. So for the most part, when people do metabolism studies, they don't really measure urine energy directly. Some will bomb urine, but most people are analyzing that urine for nitrogen and then calculating urine energy based off nitrogen content. Okay, so digestible energy minus gaseous energy minus urine energy is metabolizable energy. Okay, so then there's a fraction of that feed that's going to be lost to heat that the animal can't use for production, right? We call that heat increment. Would we expect heat increment to be higher or lower in a high forage ration versus a low forage ration? higher in high forage. So more heat production in a high forage diet as a percent of metabolizable energy intake. Okay, so ME minus heat increment gives us net energy. And so that's referred to as the usable energy source, right? So that can either be used for maintenance or growth, which is what we care about in this class, but also milk, fetal development, uh, hair production, wool, right? Make sense? Guys, remember this? Okay. Okay. Also, we're going to use metabolic body weight in this class. So metabolic body weight is shrunk body weight raised to the 3/4 power. Okay. I know you guys talked about this in feeds. Do y'all remember this? Okay. So Max Kleiber in the 40s related heat production produced per day. So this is log of heat production versus log of body weight from animals as small as a mouse to as large as a whale, and found that the slope of that line was 0.75. Okay. Whenever he did this math initially, everyone expected it to be to the 2/3 power, to the 0.67. So they assume that because as the volume of a sphere, or really any shape, increases, the surface area of that shape increases to the 2/3 power. Does that make sense? Kind of. Okay. Wizardry. So they were surprised when they found that it was to the 0.75 power. And I have yet to see a good explanation as to why it is. It is because it is. Okay. So if we take body weight, raise it to the 0.75 power, and we get metabolic body weight, that'll be important when we calculate net energy for maintenance, right? And other calculations. Okay. So the efficiency that, especially in ruminants, the efficiency that animals utilize metabolizable energy for maintenance is much higher than the efficiency of net energy for maintenance for metabolizable energy used for growth. Okay, do y'all remember that from feeds? It's somewhere about 0.7. So 70% of ME can be utilized for maintenance. Then it's somewhere around 0.5 to 0.6 for growth. The efficiency of ME utilization for maintenance versus growth. Okay. All right. So they did, Lofgren and Garrett published a paper in 1968. They were from UC Davis. They used data from 208 animals across 5 different studies. These studies had different genders, so heifers or steers, and then they also had different roughage inclusion levels. So I think the lowest was around 2% of the diet was roughage, and the highest was 100. They measured heat production, and they measured daily metabolizable energy. They regressed those against themselves. So heat production on the Y, metabolizable energy on the X, and then they solved for that intercept. Okay. So that intercept would be heat production at 0 intake or fasting heat production. So the amount of heat that's produced by an animal when they're not eating. Okay. So fasting heat production is net energy for maintenance. Okay. So they found net energy for maintenance. Importantly, they did a log heat production versus ME intake. So they did the anti-log of the intercept, and they arrived at 77 kcals per kilogram of metabolic body weight. So do you guys remember that number from feeds? Maintenance energy of beef cattle was 77 kcals per kilogram of metabolic body weight. Okay. So if we know body weight of an animal, we can take it to the 3/4 power or to the 0.75. Okay, multiply it by 0.077. That'll give us how many megacals of net energy for maintenance is needed by that animal per day. Make sense? Remember this? Any questions? You guys are gonna have to start asking questions. Okay, I'll take that as a no. All right, so NEG, older versions of the beef energy or beef cattle nutrient requirements model accounted for frame size differently than what we do current today. Okay, what you guys would have seen in feeds was a table, and you literally looked up NEG requirements for a given body weight and a given rate of gain. You guys remember doing that? Okay. Well, those numbers in that table are actually based off equations, and we're going to use the equations in this class. Okay. So, why do we need to account for differences in frame size of an animal?
Different sized animals produce different amounts of carcass, utilize energy differently than a more moderate animal, and they're gonna eat more, produce less or more off gas, equal, all that.
Like where your head's at. Cool.
Someone's gotta talk.
Yep.
It's so awkward.
Yep. It is, but I feel okay. All right. So medium frame steers have this coefficient. This is the old version. Okay. Large frame steer calves have this coefficient. So it's a larger coefficient than a medium frame steer. Okay. So this is, they used to adjust for frame size by adjusting this initial coefficient. Okay. So why did they do that? This is hard to see. Okay. But we have 1 versus 2, 3 versus 4, 5 versus 6. Okay. So a 200 kilogram medium frame steer gaining 0.6 kilograms per day would have an NEG requirement of 1.69. Okay. Let's compare that to a medium frame heifer as an NEG requirement, same body weight, same rate of gain, NEG requirement at 2.06. So about 0.4 megacals per day difference. Why would a medium frame steer require less NEG than a medium frame heifer at the same body weight, at the same rate of gain? All of these differences are because of where that animal are at in their physiological growth curve and how much, what percent of that average daily gain is fat. As a rule of thumb, it takes 5 times the amount of metabolizable energy to deposit 1 kilogram of fat as it does 1 kilogram of lean tissue. I think last year I asked this on every single one of my exams. These types of concepts. Okay. So that was steers versus heifers. Okay, 3 versus 4, 250 kilogram medium frame steer gaining 1.2 kilograms a day, needs 4.28 megacals of NEG, compared to a large frame steer gaining the same amount, 1.2 kilograms per day, needs 3.78. So a large frame steer needs less NEG than a medium frame steer at the same rate of gain, at the same body weight. And it's because of the exact same reason. At that body weight, they're at different parts of their physiological growth curve, and the medium frame steer is depositing more fat in that gain is going to require more energy. Have you guys seen the sigmoidal growth curve? Okay. So by sigmoidal, it looks like an S, right? You guys have seen this? So if this is a large, if this is a medium frame, a large frame may look like that. Okay, this is carcass composition of fat. Okay, so at 28%, empty body fat, okay, a medium frame steer maybe here, large frame steer maybe here. Make that a little more extreme. Maybe there. So it's lighter at that same body weight, at the same body composition. Make sense? We end up talking about this quite a bit because it's very important for stocker cattle, finishing cattle. Okay. So 5 verse 6, medium frame steer gaining 400, or excuse me, gaining 1.2. Oh, that's pretty similar to the last example we did. Okay, so we'll ignore that one. But we're going to talk about this again, but this is very important. Last year I had on every single test. People missed it every time. So it drove me nuts. So let's do better. Okay. So hopefully you guys gathered that it's important to adjust for frame size and physiological stage in our energy equations. Okay. So NASM now adjusts in a totally different way than what they did in the past. Instead of changing this coefficient, this 13.91, they use what's called equivalent shrunk body weight. Okay. So equivalent shrunk body weight is calculated as the shrunk body weight of that animal. Okay, so if we're formulating a ration over a whole given feeding period, that shrunk body weight we'll use will be actually average shrunk body weight. So the average body weight of the animal over the feeding period, not just at a snapshot in time. Okay, times standard reference weight divided by final shrunk body weight. Okay, standard reference weight was the weight of the animals that went into building these equations at either 25, 26.8, or 28% empty body fat. Okay, so the standard reference weight of those animals at 25% empty body fat was 435 kilograms. Okay, for a 400 or 26.8% empty body fat was 462, and then 28% empty body fat was 478. We're going to be using 478 and 28% empty body fat throughout the class. Okay. For you undergrads, I will probably give you equivalent shrunk body weight, but for graduate students, you'll need to calculate it and incorporate it into the equations. Okay. So here's an example. We buy a 700-pound steer that weighs in kilograms is 317.5. The shrunk body weight at 28% empty body fat is anticipated to be 1,240 pounds, so 564. So the equivalent shrunk body weight of that steer is 317.5 times 0.96. Okay, 0.96 times live weight is shrunk body weight. That's because most people in the world use a 4% pencil shrink. Okay, so they just take the live weight, multiply by 0.96 to apply a just straight across 4% pencil shrink. Times 478 divided by 564. So the equivalent shrunk body weight of that animal is 258.3 kilograms. Questions? All right, then we can calculate the net energy for gain that's required as 0.0635 times empty body weight to the 3/4 power times empty body gain to the 1.097. Okay, undergrads and graduate students, I'll give you these equations, and it may not even be this first exam, but you will have to use them. Okay. So empty body weight is 0.891 times shrunk body weight. So here we're going to use equivalent shrunk body weight as our shrunk body weight. Okay. So shrunk body weight is body weight times 0.96. Empty body gain is calculated as shrunk weight gain times 0.956. And then shrunk weight gain is calculated as 0.96 times average daily gain. So it seems like there's a lot of steps, but the math's pretty simple, right? What you got?
What do you, sorry, what do you mean by shrunk and empty?
Okay. So shrunk, good way to explain this. So if we take an animal and we put it in a scale, we can measure its live weight. So what that animal weighed at that given time. So if we were to hold that animal off feed and fast it for 16 hours and then get a weight, do you think it would weigh more or less?
Less.
Less. Why do you think?
It's not intaking anything, relying on.
Yeah. So it's digesting all that stuff in the rumen. Do you guys remember putting it? Do you put your hands in the cannulated animal during that feed slim?
Yeah.
The rumen is large relative to that animal's body weight. Okay, it can be as much as like 80 liters. Okay, so that's a hard thing that we try to account for in ruminant nutrition is gut fill. Okay, so standard one way we can approach that is just do a 4% pencil shrink to calculate shrunk weight gain or shrunk body weight. So we want to shrink that because if you were a buyer, do you want to pay for all that fill or do you want to pay for the actual weight of that animal?
Yeah.
Exactly, yes. So if we didn't account for this in some way, then me as a seller, I may just chunk a whole bunch of feed in front of them and make that guy pay for a bunch of rumen fill, right? Make sense?
That makes more sense.
Yeah, and so one thing is there's a difference between when you're bidding on an animal, there's a difference between live weight and pay weight, right? So we take that live weight, commonly we'll multiply by 0.96, and that'll be our pay weight or shrunk weight. Make sense? Okay, good question. All right, and so just like with shrunk body weight, we have shrunk weight gain, and we could do it initial body weight times 0.96, final body weight times 0.96, divide that all by either days on feed or days on pasture, whatever system we're looking at. That could give us shrunk body weight gain. Or we can just calculate it as live weight gain times 0.96. Okay. Empty body weight is if we took all the digest out of that animal's tract, how much would that animal weigh? So we're assuming that a shrunk animal will have 89% of its body weight that we measured shrunk would be there still. So empty body weight is an estimate of how much the animal will weigh without any digestive. Does that make sense? And so when we're calculating energy required, we need to do it on an empty body weight basis because that's the metabolizable tissue that require energy. All right. Does that make sense? Any other questions about that? Thank you for asking a question. Cool. All right. So here's an example of something that we could do if we wanted to program feed a group of animals. So we want to feed these animals a certain amount of dry matter per day to achieve a targeted rate of gain. Okay, so program fed. So we're not going to let them eat as much as they want. We're going to feed them an amount so that they will eat it and achieve a certain level of gain. Okay, so here we know type of cattle, we know the energy content of the diet, okay, and we know what our targeted level of gain is. So the question would be, how much do we need to feed them? Okay. Step one, so in this example, we have a 200 kilogram shrunk body weight, medium frame, So we'll weigh 500 kilograms at 28% empty body fat. It's a steer. So we want it to gain 1.32 pounds per day, which is 0.6 kilograms per day. Okay. It's consuming a grower ration that has 75 megacals at NEM per 100 weight. Who knows what 100 weight would be? Well, this is of the feed, right? Yeah. So 100 weight, 100 pounds. You'll see that a lot in this class. I'm always talking about pounds, 100 pounds. Okay. And this diet has 46 megacals of NEG per 100 weight, dry matter basis. Okay. So how much do we need to feed to achieve that targeted rate of gain? So step one, we need to figure out how much net energy for maintenance this animal requires. Okay. So 0.077 times 200. So we'll see there, I gave you shrunk body weight to the 3/4 power. So that animal needs 4.1 megacals per day of NEM. So step two, we want to calculate how much dry matter intake is needed just for maintenance. Because if we're just feeding towards maintenance, the animal's not going to gain anything. Also, if the animal can't gain unless we are feeding over maintenance. The animal's obviously going to use energy first to maintain its body weight instead of just for growth, right? Okay, so 4.1 is the megacals per day of NEM that's required. Divide that by 0.75. Okay, so 75 megacals per 100 weight is 0.75 megacals per pound. Okay, so that animal needs 5.47 pounds of feed just to maintain its body weight. Okay, now we need to calculate how much NEG is required. Okay, so this big long equation, 0.0635 times empty body weight to the 3/4 power times empty body gain to the 1.097. Okay, so 0.0635 times 200 times 478 divided by 500. So here we're calculating equivalent shrunk body weight. This is converting shrunk body weight to empty body weight. We're raising that to the 3/4 power. Okay. Times 0.6, so they're gaining 0.6 kilograms per day, times 0.956. So that's going from shrunk weight gain to empty body gain, okay, to the 1.097 power. Okay, so ends up being 1.62 megacals of NEG required per day. Bet you can guess what we're going to do next. Figure out how much dry matter is needed to achieve that amount of NEG. So 1.62 divided by 0.46. So that diet was 46 megacals per 100 weight. So that animal needs to consume 3.52 pounds of feed per day on top of maintenance to gain that rate of gain. Okay, so 5.47 plus 3.52 is 8.99 pounds of dry matter per day. So that animal needs to consume 8.99 pounds of dry matter per day to gain, 0.6 kilograms per day. Any questions? Okay.
Where's the energy formula from? It's not this one that's in the picture, right?
Right there. NEG required.
Oh,
Is there a different slides on Canvas?
oh, I was looking at like these, and I was like, wait a minute, it's not right.
Yeah, that's the old. Those are the old ones. Okay. So again, don't memorize these equations. I will give them to you. Work through a couple of examples. You can create your own. It's pretty easy. And apply those examples. You can make ones similar to this. We'll also have ones throughout class, but apply those numbers into these equations. Know how to implement the equation. Don't memorize the equation.
So are there different equations if it's medium frame or large?
No, not in the new one. They do that based off standard reference weight or equivalent trunk body weight. Remember me talking about that?
Yeah, I'm just trying to like understand everything.
Yeah. So in essence, we need to know not only initial body weight and final body weight, but what that animal would have weighed at 28% empty body fat. And then 478 divided by that 28% empty body fat weight times our body weight, could be average body weight, gives us equivalent trunk body weight. Does that make sense?
Yeah.
Okay. So it is adjusting for frame size because a medium frame steer is going to weigh less at 28% empty body fat than a large frame steer. So 478. So for example, 478 divided by 475 will be a larger number than 478 divided by 500. It will be a smaller number. So by making that empty body weight number, that equivalent trunk body weight number smaller, it's going to require less NEG in the equation. Does that make sense?
Yeah.
Think about what this math is doing, right? So a larger framed animal will require less NEG because it has a smaller equivalent trunk body weight that's being applied in that equation. Make sense? We'll go over this again. Okay. All right. So if we know type of cattle, we know any content of the diet, and we know how much of the diet they're consuming, we can predict what that animal should gain. Okay. Be very important in a lot of different systems to know if we have an in weight, we have a targeted out weight, we need to know rate of gain so we can calculate how many days on feed to reach our target. Does that make sense? Okay. All right, so here we have an example, 1,000-pound large frame steer. It's going to finish at 1,500. So 1,000 plus 1,500, 2,500 divided by 2 is 1,250. So its average body weight is 1,250 pounds. It's also going to weigh 1,250 pounds at 28% empty body fat. Just as a hint, your average body weight and your body weight at 28% empty body fat will not always be the same. Okay. So this animal is consuming 21 pounds per day. This diet, much higher energy, but would be pretty indicative of what we're feeding to finishing cattle. We'll have 100 megacows of NEM per hundredweight and 69 megacows of NEG per hundredweight. All that's on a dry matter basis. So what is their projected average daily gain? Okay, step one, we need to figure out how much dry matter is needed for maintenance. So we need to figure out how much NEM is required for this animal for maintenance. So that 453.5 is the initial body weight. That should really be the average body weight. So this math will be a little incorrect, but okay, we'll carry on. So NEM required, if the average body weight was that, is 7.57 megacows of NEM per day. You guys want to do this correctly, 1,250 divided by 2.205, because there's 2.205 pounds in a kilogram. That should be 566.9-ish. So raise that to the 3/4 power is 116 times 0.077. So that animal would need to consume 8.95 megacows of NEM per day to achieve maintenance. So if you want to fix those in your slides, may not be a bad idea to go ahead and do that. So that 8.95 divided by 1, because there's 1 megacal per pound, that animal would need to consume 8.95 pounds of dry matter just to achieve maintenance. Does that make sense? Any questions? You guys see where I messed up making the slide? How to fix it? Okay. So next, we need to, this is where it's going to differ from the previous one. Next, we need to determine how much dry matter intake is available for gain. So total intake is 21, should be minus that 8.95. 21 minus 8.95 is 12.05. So there's 12.05 pounds of dry matter intake that's available for gain, even though the slides here are different because I put in the wrong body weight. Guys are following? Okay. So next, we need to determine how much NEG is being eaten that is available for gain. So in the real example, it would be 12.05 times 0.69. So that animal is consuming 8.31 megacals of NEG per day that is available for gain. Make sense? All right. Final step, we 13.91 times the NEG that's available for gain to the 0.9116 power times equivalent shrunk body weight to the negative 0.6837. Okay, tell us shrunk weight gain. Here I did use the right body weight, but I used the wrong NEM or NEG. You guys see that? Okay, so 8.31, raise that up to the 0.9116 is 6.89. So instead of 7.61 here, this should be 6.89 times 13.91 times 0.0147. So that animal should be gaining 1.41 kilograms per day. These equations give it to you in kilograms. So if we want to know pounds per day, multiply that by 2.205, 3.1 pounds per day. You guys see how I did that? Any questions? No questions?
Where did you get the 6.89 from?
6.89 is the real number, which is not 9.27, but was like 8.45, raised to the 0.9116 power. That make sense? Okay. All right. So we know how to make the calculations, but in the real world, how are we measuring energy content of feed? Are we going to do a feeding trial, metabolism trial, every time we formulate a different ration? No. So we need, first, we're always going to estimate TDN. The equations that we get there are always going to be based off of nutrient analyses. Okay. The quality of the estimate is going to vary equation to equation. So we could have an equation like this. TDN is equal to 73.5 plus 0.62 times crude protein as a percent of dry matter minus 0.71 times ADF as a percent of dry matter. Okay. This equation was built for wheat pasture. Do you think an equation that's just based off of 2 nutrient analyses will be applicable across all diet types? Probably a leading question. I'm going to say no. I'm going to say that this type of equation would need to be established for every forage type, ration type. TMR will probably have a different one versus grass, which would have a different one versus hay. Not even getting into different species, so their application's pretty limited. The one I think has the most value and will be appropriate across most diets would be Weiss's summative equation. You guys won't have to use this, but I just want you to know how we get there. Okay. So Weiss's summative equation estimates digestibility of the nitrogen-free extract. So you guys would remember that from feeds, okay, as a measure of starch. In this instance, it's using nitrogen-free NDF, crude protein ash, the indigestible fraction of ash or of crude protein and ether extract times 0.98. So it's assuming that 98% of that nitrogen-free extract is digestible, plus KD is an estimate of crude protein digestibility times crude protein, plus 2.25 times ether extract, minus 10. So that minus 10 is trying to account for any plant waxes that end up in ether extract, plus 0.75 times nitrogen-free NDF minus lignin times this. In essence, it's trying to estimate a digestibility coefficient for introdetergent fiber, and then minus 7. So whenever I played around with this, it works. This equation works really well. Again, I'm not going to ask you guys to use it. Okay. So you don't need to know. I may ask you, where would you find the summative equation to estimate TDN? Answer would be Weiss 1992 or something like that. Okay. All right. Once we know TDN, we can get to DE. So 4.401 divided by 100 times TDN as a percent of dry matter gives us DE. And then how we can go from DE to ME, there's several different options. The most up-to-date one that I imagine will be in the newest update of beef cattle nutrient requirements are either this Galleon from 2016 or this Hales from this Hales paper in 2022, would be my guess. They give you very similar estimates. Okay. So then once we know ME, we can go to NEM and NEG using these cubic equations. So this is what's currently in the beef cattle nutrient requirements model. These are the equations that are suggested to be used if you're calculating ME using Galleon or the Hales equation. So again, I'm not going to ask you to even use these equations. I just want you to know how we get there. Okay? So important to know, most of the time what people are using to formulate rations are mathematically derived from TDN. Okay? So why don't we make equations that go directly from TDN to shrunk weight gain? I don't know. They like to be complicated. Probably it is a little easier to account for the differences in efficiency of ME utilization for net energy for maintenance versus gain. Probably. But other countries do just go directly from ME to shrunk weight gain, right? They don't worry about NEM and NEG. They apply that efficiency of ME utilization directly to that ME content of the diet. Make sense? So welcome to America. Okay. Any questions? All right. Crude protein. 2 real fates of nitrogen that's consumed by the animal. It's either digested in the fermented in the rumen, and it's rumen degradable protein, or it's not, and it's rumen undegradable protein. Okay. So that RDP can either be incorporated into microbial crude protein, so utilized by the microbes in the rumen to allow them to replicate, to make amino acids, and then that animal can use those microbial crude protein once it gets digested in the small intestine to support the metabolizable protein needs of the animal. Okay. Or it's not fermented in the rumen, it's rumen undegradable protein. That rumen undegradable protein will go to the small intestine to be potentially metabolized by your proteases secreted from the pancreas. Those amino acids are absorbed in the small intestine to be used as metabolizable protein for the animal. Okay? So not all of that RUP will be metabolized in the small intestine. So we need to know to get to MP, we need to know a digestibility coefficient of that RUP. Okay? This is all new. Okay. Again, this is kind of an introduction. We will probably talk about this some further. So some of this protein stuff is going to be a little more information what you need. Okay. All right. So that RDP that's converted, some of it's going to be captured as MCP, some of it's going to be lost, absorbed as ammonia from the rumen wall. That ammonia is going to go to the bloodstream, going to be gone to the liver, going to be converted to urea, and then excreted in urine, urea, nitrogen. Okay. So the amount of that ammonia that is captured by those microbes to be converted to MCP is going to be based off of how much available carbon there is for them to match carbon skeleton with ammonia. I know you guys went over this concept in feeds and feeding. Okay. So we need to calculate a TDN allowable MCP. Okay. If we're talking about roughage diets or diets that have less than 4% of ether extract, we can just use total TDN. If we're talking about diets that are really high in fat, we need to use fat-free TDN. Okay. Why would we need to use fat-free TDN if the ration has a whole lot of fat? What happens to lipids when they're consumed by the ruminant? Can microbes ferment fatty acids? What do microbes do to unsaturated fatty acids? So biohydrogenation, that's the extent of metabolism. They can cleave them off of glycerol backbones. They can use that glycerol, but they're not actually using that fatty acid for energy. The extent of a change that can happen to that fatty acid is biohydrogenation, where they're breaking double bonds and slapping hydrogens to those fatty acids. Okay? So if that carbon from that fat, that ether extract, is not able to be used by the MCP for MCP synthesis, and that ether extract has a large percent of the diet, right, then we need to account for that, take it out of the TDN when we're calculating TDN allowable MCP. Does that make sense? Okay. So if we know TDN allowable MCP and we know RDP, so how much of that protein is fermented in the rumen, we can calculate, what's that here? Oh, here we go. So we can measure RDP in several different ways, okay? So this was taken from in situ digestions. You guys did that in feeds. You put your bags in for one time point. They took it out of the rumen. They measured how much was lost from the bag. You measured in situ dry matter digestibility in feeds and feeding, right? So if we do a similar approach, but we have increasing time points, okay, you end up with a shape like this, okay? We can fit that to the Orskov model, and we can calculate an A fraction which would be the intercept, okay? Soluble crude protein, okay? So how much crude protein was digested at hour 0, so no room and incubation time, okay? We can calculate a B fraction, which is related to the asymptotic CP digestion. So who knows what the asymptote is?
The maximum.
The potential, right? Because it's never actually going to get there. So that represents the non-soluble fraction of crude protein that could be potentially digested in the rumen. Okay, we compare that with rate of digestion and rate of passage, and we can calculate RDP. So the soluble fraction, we assume that all of that 100% of the soluble fraction is RDP plus B, so our asymptotic crude protein digestibility, times rate of digestion divided by rate of digestion plus rate of passage, okay, will give us RDP as a percent of crude protein. Okay, you won't have to know that. I just want to make sure you guys at least have somewhere to go back to understand where RDP is, how we measure it. Okay. Cool. So we can measure RDP using in situ approaches, or we can use in vitro approaches. So we take rumen fluid out of the cannulated animal, we incubate samples in that rumen fluid in a jar for a period of time. We measure how much crude protein was digested. Could be another way, probably less accurate. And there's another in vitro approach where we take Streptomyces rhesus and we incubate that with the samples. And those Streptomyces rhesus is supplying protease to that solution to break down proteins. So that's another in vitro procedure we can take. However, again, probably pretty inaccurate. Then more recently, my PhD student there, when I was still at Bushland, developed this equation for RDP, used 18 different ingredients, did in situ digestibilities. That has not been published yet, but we found that we could very accurately predict RDP based off NDF content and crude protein. So now we're getting to a point where maybe that's actually usable by producers, right? Because producers aren't going to incubate their samples in a rumen for 72 hours and measure rate of digestion. Okay. So most people are formulating rations. If they're doing anything other than crude protein basis, they're doing it based off book values for RDP. Okay. So you guys remember TDN allowable MCP? So if we know RDP intake, so how much total RDP the animal consumed, we know TDN allowable MCP, we can calculate RDP balance. Okay, if RDP balance is positive, so if there's more RDP intake than there is TDN allowable MCP yield, then RDP is equal to TDN allowable MCP, or excuse me, MCP yield, how much microbial crude protein that will be produced will be equal to how much crude protein or how much MCP is allowed by the TDN content. If it's negative, then the MCP yield is equal to RDP allowable MCP. Okay, questions? Okay, so then if we know MCP yield and we know a digestibility coefficient of RUP, we know RUP content, we can calculate metabolizable protein. So metabolizable protein that's being supplied to the animal is 0.64 times MCP yield plus the digestibility RUP coefficient times RUP. So that 0.64 times MCP yield is because we assume no one's really measured this directly. It's just a best guess that 80% of MCP is true protein And so about 20% of that microbial crude protein is nucleic acids. Okay. And then only 80% of that true protein will be digestible. So 0.8 times 0.8 is 0.64. Does that make sense? Okay. All right. So most people are formulating rations on a crude protein basis because to get accurate estimates of MP is very difficult. Okay. Any questions? All right. This is just going to go over how to calculate RUP. So RUP is 100 minus RDP. So then we can measure the digestible coefficient for RUP. So what percent of that RUP is digestible? A few different techniques exist. So we can use the mobile bag technique that takes a duodenal cannulated animal. So where the small intestine is cannulated. You guys saw a rumen cannula. This is a cannula in the duodenum in the small intestine. They insert filter bags, similar to F-57s, into the duodenum, allow it to go through the intestinal tract, go through all the different enzymatic digestions that occur in the small intestine. Then they collect those bags as they get excreted in feces and measure the digestible crude protein from that. So pretty obvious why not a lot of people do that. Very expensive. Duodenal cannulated cattle don't last very long. So you have all that sunk cost of the animal. It's very labor intensive. Okay. Another approach, they take a cicotomized rooster, okay, chicken, and then put basically a cannula into the cecum, put filter bags in there, incubate it in that cicotomized rooster for a period of time, take it out, measure how much crude protein of that RUP was digested. Okay, cheaper, chickens are cheaper, still labor intensive. Cicotomized roosters don't last very long. Okay. Then there's a step that I've done, in vitro step. It's called a 3-step procedure. Step 1, you incubate samples just like you would in C2 in the rumen. Do it for about 12 to 16 hours. Then you take that out. You do a HCl and pepsin digestion in the lab. That simulates the true stomach digestion of those samples. Then you take those samples and you incubate them in pancreatin, thymol, and then a phosphate buffer. Do that for 24 hours, and you can measure how much of that RUP was digested from there. So that's kind of an in vitro technique to stimulate small intestine digestion.
Cost-wise, relatively, where do they go?
The mobile bag technique is going to be the most expensive. Cicotomized rooster is going to be the second. And the cheapest will be the in vitro modified 3-step procedure. Okay. So that's how we can directly measure it. The current NASM, hopefully this gets updated with my PhD students' research in the future, if it ever gets published. I was just on this committee. I worked for the USDA, was on a committee of a graduate student out there, but spent a lot of hours collecting these samples. So the NASM just assumes a digestible RUP coefficient of 60% for roughages and 80% for concentrates. Doesn't seem very accurate. And when we did the modified 3-step procedure for Jarrett's dissertation and saw that DRUP ranged from 0.4 to 0.9, right? Huge range based off of type of ingredient that these assumed fixed coefficients aren't capturing in the current model. Okay. So we also found that we could predict digestible RUP as 0.8 times RUP minus 0.02 times NDF content. Maybe that'll get adopted in the new NASM. I don't know. Okay. You guys have used calculator in feeds before. We're gonna use it a lot more. Okay. And we're going to be balancing not just calcium and phosphorus, but all the macro and micro minerals that are included in it. We're also going to use it to formulate premixes. Okay, we're going to use it to formulate premixes that are medicated. Okay, these would be type B medicated feeds that we'll use this for. There's some tricks that I'll show you how to make it a little easier. There are lab assignments where you'll use this. You will not use calculator for your exams. Okay, but will be used for your lab assignments. Are you guys done for the day? Okay. We can go over it at a later time, unless you guys want to do it now. All right, what's your homework assignment for tomorrow? Bring this back. Okay. Cool, guys.
The next week.
Let's have it tomorrow
For the class.
for the class. Yeah. Yeah. Because based off what you choose, we're going to give you assigned first, second, third treatment goals, percents, and decimals. And then we'll start making the calculations for our enterprise budget.
If I could ask you for
Sure.
Yeah, of course. Okay, so I have never taken ever. I am a ecosystems background in environmental science and soil science.
Okay.
I have like my whole project in beef was meat science with environmental science. So I never touched any of the beef background.
Yep.
And so I'm an super lab professor and I'm trying to decide, is this something that's going to require me to like take feeds and eat it on my own at this point?
No.
Because it's just like you're Victoria's pig rat in a lot of ways. Like I know the basic terms,
Okay.
but I haven't gone past it.
Yeah. From this lecture, you're really going to need to know what the concepts are and then how to make those calculations for energy.
Because I'm like, if this is gonna be one of those things where it's like if you don't know feeds, you're kind of a Greek.
People have taken it without feeds and feeding before
Okay.
and been fine.
Because I'm like, this is the week to make that choice. If
Yeah.
so I just wanted to like see if this is a like, yes, you're gonna be fine.
If you can read an equation and then the word problem where it says this is your shrunk body weight, plug that into your
Okay.
equation.
Because I'm like, this is my first time seeing these equations, so we're
Well,
really
good.
talking about it. I'm like, this is awesome because I kind of understand the other side, how the other side lives.
Yeah.
But because I've never touched it and I know I'm spending 10 hours a day in a lab this semester, I'm like, I just wanted to clarify, this is a thing where it's all.
From this lab, I would expect you to know terms. Okay. I would expect you to know how to implement the equations,
Yes, sir.
mainly around energy. And so NEM, MEG needed for a given level of intake or for a given level of average. Okay. Then if I ask you what is RDP, I would expect you to, if I ask you to tell me what RDP is, I would expect you to know how to do that.
Like room integrated.
Yeah, room integrated protein.
Yeah, like, because all I work with
Yeah.
I'm like sitting in here and just thinking, yeah, this is a no, since I'm not animal science, I'm like,
Yeah. People have taken it outside of the past, don't need to be in there.
and they don't have like unit tests.
No.
Okay. Because I'm like, I'm just thinking along that because I know what this semester looks like for me, but if you think it'll be good, I'm not gonna worry.
You'll be fine.
Okay.
Yeah.
Because, yeah, that's a lot.
I would say 60% of it's econ stuff.
Okay, which I also have never taken.
And you're very close to everybody else. Yeah, you'll be fine.
Perfect.
Cool. Okay. There was another, so I think there's 4 people missing.
Okay.
So we'll probably lump them together. Okay. So we'll get you teamed up with, yeah.
Yeah.
Okay, we will get you in touch. We'll do that tomorrow. Yes, find me first thing tomorrow at lecture, and we'll get you guys in touch. We can probably decide that after or before lecture. Yep.
Yeah. And that probably is one of those questions that you're like, well, of course I work in science, like whatever. I just, being a soil scientist comes with different